And so we now move on to the next speaker, Dr. Allan Fernando, a colleague from the National Institute of Geological Sciences. Dr. Fernando is now professor at NIGS and actually is a returning deputy director for academic affairs by tomorrow, having finished his sabbatical after Dr. Jenny Ramos ably took over for him for a while. Dr. Fernando obtained his BS and MS in geology at UP and his PhD degree in Hokkaido University in 2006, after which he joined NIGS as assistant professor. He also has a diploma in archaeology from the Archaeological Studies program in 2015. Allan studies calcareous nanofossil biostratigraphy and has been involved in the study of sedimentary basins in the Philippines in collaboration with the Mines and Geosciences Bureau and the National Museum of Nature and Science in Tokyo, Japan. He has authored and co-authored several papers that dealt mostly with the use of nanofossils as biostratigraphic and paleoceanographic tools from the Cretaceous to recent. He is currently involved in research projects on the Cenozoic mollusk diversity in the Philippines and the EIDR program, Ma'am Giselle would be happy, on discovering the world of the very first hominins in the Philippines, geology, paleoenvironment, and paleoecology of archaic hominins in the Philippine Islands, Project 1 Geological Environments. Dr. Fernando's presentation today will focus on the newly discovered fossil localities from these projects. Allan, please take it away.
So can you hear me?
Yes.
Okay, so I'm going to start screen sharing.
Yeah, we see it.
We see it already.
Slideshow na lang.
Okay, so good morning to everyone, and thank you very much for the introduction. So I'm Allan Fernando, and I'm from UP NIGS, and my presentation today reports new localities in the Philippines that yielded interesting fossils that we hope to contribute something or a couple of things to the current knowledge on Philippine paleofauna. Before I begin my presentation proper, I would like to acknowledge my co-authors and all the people who have contributed to this presentation. So we all know that present-day Philippines is considered one of the megadiverse countries in the world. And one way to illustrate this is actually to show maps that compare the Philippines with other countries in terms of terrestrial and marine flora and fauna diversity. And based on several studies that have been done so far, the Philippines is considered the country with one of the highest diversities in marine organisms, as well as one of the countries with the highest number of endemic mammals in the world on a per area basis. And knowing this information, one might ask, were we also as biodiverse in the geologic past as we are at present? And to be able to answer this question, we actually had to look for clues in the fossil record, which can provide us information regarding how the Philippines became the center of biodiversity and how the country's unique marine and terrestrial flora and fauna developed. And with the most recent discoveries of fossils in the Philippines in the last few years, there's quite a few during the last 2 years, there seems to be a renewed interest in paleontology in the country. And this renewed interest actually represents both an opportunity and a challenge for us paleontologists, which we hope to address in the years to come. So one of the main motivations of this presentation is actually to let people know that paleontological studies are very much alive in the Philippines, despite the fact that we study long dead remains of plants and animals that have been already preserved in the rock record. Another motivation is actually to educate people or let people know that collecting fossils without proper documentation is actually not the way to do or to conduct a proper paleontological research. Since when you remove a fossil from the site, it diminishes their scientific value and actually deprives the Filipino people of any important scientific information that we paleontologists can gather from them. So at present, there are several institutions and agencies that are actively involved in paleontological research, and these include the MIGS and the Archaeological Studies Program in UP Diliman, the Mines and Geosciences Bureau, and the National Museum of the Philippines. And then, of course, we also have the Paleontological Society of the Philippines, which was relaunched in 2019 and is currently headed by Dr. Peleo Alampay, who is one of the hosts of this parallel session. So PALEOSOC aims to promote and to develop branches of paleontology in the Philippines. And if you want to know more about the Paleontological Society of the Philippines, you can visit our Facebook page and click the like button. So for the outline of my presentation, I would like to show you these two maps that basically reports the location of sites where vertebrate and chemosynthetic mollusk localities in the Philippines are reported. So the figure on the left shows the map of the different vertebrate localities in the Philippines, while the map on the right shows the two mollusk localities in the Philippines that are chemosynthetic in origin. So my presentation is divided into three parts or three sections, the first two of which will actually deal with the new vertebrate localities in La Union and Palawan, while the third part of my presentation will discuss the chemosynthetic mollusk assemblage in the province of Pangasinan. And what I will do is at the end of each presentation, I will try to discuss the implications of the fossil finds to the current understanding of that particular fossil group or fossil groups in the Philippines, and if applicable, their implications also in the West Pacific region. So most of the studies that I'm going to present today are still ongoing, so all the data that I will present are considered preliminary data. Sorry for that. So the first locality that I would like to discuss is located in the province of La Union, which is located in the Ilocos Central Luzon Basin, which is one of the major sedimentary basins recognized in the Philippines. The basin is filled up with sediments deposited in a wide range of marine environments during the Oligocene to the Pleistocene interval. The youngest sedimentary units in the basin, where the vertebrate fossils are discovered both in Pangasinan and in La Union, are actually deposited in shallow marine to terrestrial environments. And before the discovery of our site in Bacnotan, La Union, the only known vertebrate locality in the basin is located in Anda, Pangasinan, which is shown in this yellow circle here, where remains of stegodon, elephant, rhinoceros, deer, and other vertebrate remains are reported. An example of which is this picture of an elephant fossil, which is actually in the private collection of a family in Anda when we conducted our fieldwork in the area last year. So for the fossil locality in La Union, please take your attention to this red box here in the geologic map of La Union, which is the municipality of Bacnotan. So Bacnotan is a municipality in La Union, which is located north of the city of San Fernando. So these figures here show actually the geologic map of La Union and the stratigraphic column of the province, which shows the succession of rock units in the area, showing that the Mortis Formation, which you can see in this upper part of the stratigraphic column, is the youngest unit in the area. So the rocks that we have in Bacnotan is actually underlain by Bacnotan limestone, which is underlain or which is considered a facies of the Mortis Formation, which, as I had mentioned earlier, is the youngest sedimentary unit in the province. Within the Bacnotan limestone, the fossil locality is situated in an ancient channel deposit that is interpreted to have been deposited in a fluvial deltaic depositional environment based on available sedimentological and paleontological data. And this is a picture of the outcrop where the fossils were collected, showing clearly the channel that eroded through the underlying limestone unit, which is the Baknotan limestone. It's not very clear in this picture, but actually the base of the channel deposit is composed of fragmented parts of the Baknotan limestone, suggesting erosion of the underlying limestone unit before the deposition of the interbedded sandstones and siltstones, as well as conglomerate lenses, which comprise these channel deposits. So in the channel deposit, the fossils were collected near the upper and lower parts of the channel within the sandstone units. And grain size analyses of the sandstones reveal the dominance of several components, including quartz and some lytic fragments, which were identified as PAASE fragments, suggesting that they are volcanic in origin. So a picture of the composition of the sediment is shown in this part, showing you clearly that quartz is the dominant component of the sandstone unit. And quartz, by the way, comprise 70% of the sandstone assemblage, or the sandstone component. Other components that we observe in the sandstone include magnetite and planktonic and benthic foraminifera, which were used to interpret and support the deposition of the channel took place in the marine environment. We still haven't dated the channel deposits, but based on the age of the underlying limestone, which is the Baknotan limestone, we consider the fossils Pleistocene in age. The source of the volcanic materials comprising the channel deposits, however, has not yet been determined. And actually, one of my students was going to enroll in the master's program of MIGS, will be the one to study this outcrop and hopefully be able to identify the source of the volcanic materials when he actually does the paleo environment reconstruction of the site during the Pleistocene epoch, which will be part of his master's thesis. And based on the analyses and the samples we have collected so far from the Baknotan outcrop, fossils we have include cervids, bovids, land tortoise, and crocodile, which I will be showing in the next few slides. So these are the cervid teeth fossils. Cervid includes deer, which were collected during the 2019-2020 fieldwork by MIGS and ASP under the EIDR program. So prior to the collection or prior to the fieldwork, which resulted to the collection of these teeth fossils, only 3 specimens were collected from the Baknotan site. Of these 3 bone fragments, 2 are cervid humerus bones, and 1 is a land tortoise bone, which are actually on display at the MIGS Uggaa Museum. So at the time when these 3 bone fragments were collected in the Baknotan area, the source of the fossils have not yet been identified, since the fossils, the bone fragments, were found as floats and are already separated from the matrix. And lucky for us, last year in 2019, we were able to finally find the source where the fossils are coming from, which is the channel deposit, which was eventually followed by a more detailed description of the channel, more detailed description of its extent, and a more systematic collection of samples which will be used for dating, grain size analysis, and of course, the taxonomic identification of the fossils that we found and we will find in the future within the channel deposit. So one student who is involved in the project is actually doing a preliminary analysis of the bones, the cervid bones that we collected from the site earlier. And what is shown here are the pictures of the bones, which were subjected to morphometric and feature analysis by this master student. And based on his analysis, the bones probably belong to a large, unclassified extinct species of deer with a mixture of characteristics from the Timor deer, which can only be found in the Indonesian archipelago, and the Philippine brown deer, which of course is endemic to the Philippines. Although this analysis is preliminary, it is very important because the 2 species are endemic to areas that are already far from each other, suggesting that they definitely have something to do or tells us something about how this extinct species migrated from one place to another place. Based on the available data that we have for the Baknotan site, the bones that we found in Baknotan is the possible ancestor of this Timor deer, which can only be found in the Indonesian archipelago. So it is possible that this extinct species of deer that was present in Baknotan migrated southwards to the Indonesian archipelago and eventually evolved into a smaller, different species of deer after its isolation. So we need more data, we need more specimens from the site in order for us to support this initial model or initial hypothesis. The next slide shows the fossils of the Bovibi teeth, which were also collected during the 2019-2020 fieldwork by NICS and ASP. And similar to the cervid bones and teeth that were shown in the earlier slides, analyses of the bovid fossils are still ongoing. So we still don't know whether they belong to a new species or to something that has already been described. But if it's going to be a new species, it's going to be a great contribution to the Pleistocene vertebrate fauna inventory of the Philippines, given that the last described extinct species of bovid in the country was in 2006, when the Cebu dwarf buffalo, or Bubalus cebuensis, was formally described from Cebu Island. And a figure on the lower right of this slide shows the reconstruction of what the Cebu dwarf buffalo looks like in comparison with the Philippine carabao and the tamarao, which can only be found in Mindoro Island. So more analysis of the bovid bones will be done later when we collect enough specimens to do a more detailed taxonomic description and analysis. And lastly, we have this crocodile tooth that was also recovered from the channel deposit in Baknotan, La Union, which is actually very large, approximately 3 inches in length. Although crocodile bones have already been described and reported from the Cagayan Valley Basin, correct me if I'm wrong for those of you who are actually working at the National Museum, but this crocodile tooth is probably the first crocodile tooth fossil found in the Philippines. And based on its large size, it's approximately 3 inches in length, the tooth could have actually come from a saltwater crocodile or Indo-Pacific water or Indo-Pacific crocodile, also known as Crocodilus porosus, which, as you can see in this figure on the left, evolved sometime during the Pliocene epoch. This figure on the left, by the way, is from the paper of Broku in 1997, showing the stratigraphic distribution of the different Crocodilus species. And you can see clearly in this figure that Crocodilus porosus evolved during the Pliocene epoch. If our identification is confirmed, then this could be the earliest record of Crocodilus porosus in the Philippines. So aside from crocodile tooth, we also have some reptile bones recovered from the site as well, but we have yet to determine their taxonomic affinity, whether they belong to the same specimen where the tooth came from, or whether they belong to a separate reptile or a separate individual that's also present in the Baknotan site. So right now, we are working with Crocodilus porosus Incorporated through Sir Rainier Manalo in an attempt to reconstruct the size of the crocodile where this tooth came from, which is going to be based on the tooth-skull size ratio, which we hope to do after the pandemic. But comparing the size of this tooth with the size of the teeth from the present-day Crocodilus porosus, probably we are dealing with a crocodile that is probably as large as Lolong, which, as we know, is the largest crocodile in captivity until its demise a few years ago. And I think just an additional note, in a separate fieldwork that was conducted also last year, we also found a smaller crocodile tooth fossil in Isabela, which is older than the crocodile tooth that we found in Baknotan. And the age of that crocodile tooth, which is also smaller in size, is late Miocene in age. So from here, we go to the next site, which is Palawan. So we all know that for a very long time, Palawan has been synonymous with Tabon men. which was then the earliest human remains in the Philippines until the discovery of the Homo luzinensis in the Cagayan Valley Basin, particularly in Calao Cave. In 2011, Palawan made headlines again when a sirenia, or a dugong fossil, was discovered inside the Puerto Princesa underground river, which is indicated in this geologic map by this red circle here. The Sirenian fossil is found embedded in a limestone that belongs to the late Oligocene to the early Miocene St. Paul limestone, which is best exposed along the road leading to Sabuang, Puerto Princesa, which is the area enclosed in this blue box here. In 2016, a local caving group and the Palawan Council for Sustainable Development reported the occurrence of fossils in one of the tower karsts in Puerto Princesa City, south of the Sirenia locality. And this locality is indicated by this red star located in Barangay Tagabinet. So after the report of the caving group and PCSD, a reconnaissance fieldwork was immediately conducted to assess the site and to document the fossils inside the cave. Due to the fragile nature of the bones and the difficulty in removing them from the surrounding matrix, which is really hard, we decided to leave the fossils intact and plan our next fieldwork. But this time we're going to be joined by experts in this particular fossil group coming from Taiwan and of course the Philippines. And lucky for us that the site is well protected by the locals, is under the strict management of PCSD, ensuring that the fossils will be safe until we conduct our detailed study, hopefully after the pandemic. This is the tower karst where the fossils were discovered. So the fossils are situated approximately 25 meters from the forest floor and can only be accessed through rappelling. And the fossils are located further from the main entrance of the cave and can be accessed by passing through cracks and holes that are big enough to allow just one person to pass through. So it's really difficult to access the site, which is really good because that would prevent people from illegally collecting the fossils before we conduct our detailed study. And what do the fossils look like? So these are the fossils that were found inside the cave, inside the karst tower, which were identified to belong to a cetacean, a group of animals that includes dolphins and whales. Unlike the Sirenian fossil, however, which was found within the limestone, the cetacean fossils were observed embedded within sediment-filled fractures within the limestone, which is shown very clear in these pictures as the yellowish brown material surrounding the different skeletal elements, which is in great contrast to the color of the limestone in some of the pictures here, which are light or white in color. So what's the implication of finding these cetacean fossils inside the sediment-filled fractures? This suggests that the cetacean fossils are younger than early Miocene. They're younger than 16 million years old and is therefore younger than the dugong or sirenia fossil discovered in the underground river. Although one problem right now is that we don't have any datable material at present, which prevents us in having a more accurate determination of its age, which again, hopefully, we will be able to do after the pandemic, after we have collected the bones, and after we have collected the matrices for possible dating of the fossils and the sediments within the fractures. So preliminary identification of the bones present inside the karst tower suggests the presence of different skeletal elements, including thoracic vertebra, possible ribs, limb, long bones, and even a humeral bone or a humerus. Many other skeletal elements are present, but we cannot identify them with certainty unless we remove them from the matrix, which we hope to do very soon. And for those of you asking or who's going to ask about the preservation of the bones, they are very well preserved because you can see that the internal structures of the bones are still visible, and the bones show very minimal evidence of permineralization and replacement. And these characteristics of the cetacean bones inside the karst tower is actually another supporting evidence that indeed the cetacean bones are definitely younger than the surrounding limestone unit, which is of course the St. Paul limestone. So why is this fossil find important? Well, aside from being the first cetacean fossil record in the Philippines, the fossils in addition represent an opportunity to provide information regarding cetacean evolution and dispersion in the West Pacific region. So if you're going to look at this map where cetacean fossils have been reported, you can see that the West Pacific region is data deficient, with the exception of Taiwan and Japan, where most data are actually coming from. Therefore, if we are going to conduct a detailed study of the cetacean fossils in Palawan, it will definitely help in elucidating the evolutionary history of the dolphins in the West Pacific, as well as add knowledge to the aquatic vertebrate paleofauna of the Philippines, which are rarely discussed and studied in the country, despite the fact that the Philippines has a lot of sedimentary rocks that were deposited in the marine environment. So we have to take note that prior to the discovery of the Sirenia and cetacean fossils in Palawan, much of what is known about the vertebrate fossil record in the Philippines are from terrestrial vertebrates. And I think it's high time that we focus our attention to this important group of animals, which again can provide insights how the Philippines became a center of biodiversity with respect to these marine organisms. So for the last part of my presentation, which is on the chemosynthetic mollusk locality in Pangasinan, I'd like to acknowledge my student, so Clarence Magtoto, who is actually the one who conducted the detailed study of this site in Pangasinan. So he is now preparing a paper regarding the geology and the taxonomy of the chemosynthetic bivalves that we discovered in this particular site. But before I show you what information we have so far, let me just give you a brief background on chemosynthetic habitats and the process of chemosynthesis. So environments with little or no photosynthetic activity and instead have high sulfur and or methane production levels are usually associated with chemosynthetic habitats, examples of which include cold seeps, methane seeps, hydrothermal vents, and even sunken organic remains, which are very common in the open marine environment. Chemosynthetic habitats can also be found in environments with relatively high photosynthetic activity, such as estuaries, mangrove swamps, and even seagrass beds. The main process used by some organisms in these chemosynthetic habitats to convert inorganic matter into organic matter is through the process known as chemosynthesis, which relies on energy sources such as elemental sulfur, hydrogen sulfide, molecular hydrogen, and so on. And among the organisms that are common in the fossil record associated with chemosynthetic habitats are the chemosynthetic bivalves. And what are these chemosynthetic bivalves? These are mollusks that survive through the hosting of chemoautotropic bacteria in their tissues for energy production and carbon fixation. And in the Philippines, the first documented occurrence of chemosynthetic mollusks is in Leyte, where a recent study, which is going to be published very soon, supports the important role of the Philippines in generating biodiversity in the Indo-Pacific region with respect to chemosynthetic bivalves, and which also actually strengthens an earlier idea that the Philippines is a center of biodiversity, specifically for some families of this peculiar mollusk, like for example, for the family Lucinidae. So in this study that was conducted in Leyte, they were able to identify more than 20 new species of these chemosynthetic bivalves, which again suggests that the Philippines is indeed a center of biodiversity for this particular group of marine organisms. In Pangasinan, the chemosynthetic mollusk locality is located in the Northern Cement Corporation quarry in Barangay Labayug in Sison. And these two figures shown in the map are the geologic map of the quarry area and an example of an outcrop of the late Miocene Amlang Formation where the chemosynthetic mollusks were observed. So where in the Amlang Formation, the chemosynthetic bivalves were observed, they were observed in blocks, in rock fragments that are incorporated within the conglomerate layers of the Amlang Formation. And using planktonic foraminifera, the blocks were dated as early Miocene, suggesting that they were reworked along with the chemosynthetic bivalves from an older sedimentary unit that eventually was incorporated within the late Miocene Amlang Formation. And these are some of the pictures of the blocks that were observed in the different stratigraphic levels within the quarry area, which, as you can see, are composed of indurated dark gray siltstone and sandstone units, and dense accumulation of mollusks, which are represented in these blocks by these white materials here, which we were able to separate from the matrix in order for us to do taxonomic analysis. And in order for our colleagues from Sweden and Japan to do identification, what mollusks are present in these blocks, which are very common in the area. The matrices of the blocks, by the way, were also subjected to carbon isotope analysis in order for us to infer the depositional environment of this chemosynthetic mollusk. So this is the picture of the mollusk after they were separated from the blocks. So some of them were separated using a micro drill or a dental drill, because as I have said earlier, the blocks are very indurated. So we really have to use not really sophisticated equipments, but we need to use a lot of things in order for us to separate these bivalves. And based on the analysis of our colleagues from the National Museum of Nature and Science in Tokyo and from the Swedish Natural History Museum, they were able to identify that the chemosynthetic mollusk assemblage in Pangasinan consists of 6 families of chemosynthetic bivalves, 14 genera, of which 1 is a new genus, which will be named after Season Pangasinan, and 19 species, of which 8 or possibly more are also new to science. They were also able to identify a lot of gastropod species in the assemblage, of which 1 is also a new gastropod species. So these bivalves and these gastropods, they form what we call the Miocene cold seep molluscan assemblage in Season, Pangasinan. So based on the known habitat preference of the chemosynthetic mollusk, and based on the carbon isotope analysis and the values measured from the matrices of the blocks, it is inferred that the fossils represent a cold seep assemblage. So it's either a methane seep or an environment that actually experiences a lot of organic decay. And based on the age of the blocks, which is early Miocene, this assemblage is also so far considered the oldest chemosynthetic mollusk fossils in the Philippines. Because if you're going to compare this with the mollusks that we discovered also in Leyte, the mollusks are only Pliocene and Pleistocene in age, which is definitely younger than the early Miocene that we identified for the mollusks in Pangasinan. So the big question now is, where are the blocks coming from? So we are looking at the possibility of zigzag formation as the possible source of the blocks based on the age. But looking at previous studies that actually concerns age of the rocks that we have in the Ilocos Central Luzon Basin, no such fossils were reported from the formation in other in the area. So we are still looking at the possibility that the blocks could have been sourced elsewhere, could have been sourced from another sedimentary unit, but at present, we don't have any answer at all. So that's, again, another future study or another study that we will do in the very next feature. And based on the number of the new species that we recovered from the site, this study also supports the model or the earlier idea that the Philippines is indeed a biodiversity hotspot with respect to chemosynthetic mollusks. So for future plans, so we still need to do a lot of analyses. We still need to do a lot of fossil collection. But of course, the ultimate plan is to publish our results and again, be able to contribute to the understanding of the Philippine vertebrate and molluscan paleofauna and inspire, of course, a lot of people to pursue a career in paleontology, not necessarily vertebrate paleontology, but paleontology in general. So thank you very much.
Thank you very much, Allan. The question and answer portion will be after the next talk. And so we will reserve it for that. So in the meantime, maybe Dr. Fernando, you can view some of the questions already and get ready for them after the next talk, which may I turn over the floor to Dr. Concepcion. Giselle, are you there?
Yes, yes. Thank you very much, Dr. Alisa.